Hello, you are listening to 1 and 2, a Manchester Cancer Research podcast, brought to you by the University of Manchester and the Manchester Cancer Research Centre. With 1 in 2 of us receiving a cancer diagnosis at some point in our lifetime, it has never been more important for our research to improve the outcomes of people affected by cancer. I am your host, Sally Best, and throughout this series I will be speaking with Manchester cancer researchers about their innovations, discoveries and projects that are changing the landscape of lung cancer detection and treatment. Lung cancer is the leading cause of cancer death worldwide, causing 35,000 deaths in the UK each year alone. As such, it is one of CRUK's 4 cancers of unmet need. Of 47,000 new lung cancer cases every year in the UK, 15% of those diagnosed are never smokers. Here in Manchester, we have one of the worst lung cancer mortality rates in England, so we are striving to improve early detection and treatment of this cancer. Across these episodes, we explore lung cancer from the genetics and diagnoses to screening and treatment, talking to Manchester researchers who specialise in basic, translational and clinical lung cancer research. And remember, you only need lungs to get lung cancer. In this episode, we speak to Dr Colin Lindsay about the basic biology of lung cancer, focusing on the high incidence of lung cancer here in Manchester, the differences between non-small cell and small cell lung cancer, and oncogenic drivers in lung cancer such as the KRAS mutation. He also discusses his work on the drug sotorasib, which targets KRAS mutant cancer and has now been approved by the FDA and for use in the UK by NICE. Hello everybody! Thanks for joining us for our first episode of season 2, which is focusing on lung cancer. Very exciting. I'm really excited to be hosting this series and talking to an amazing and erudite selection of people, including the one and only Colin Lindsay. God, we're already at season 2. How are you feeling?
It's a pleasure to be here. What can I say? Thank you, and a privilege.
A privilege, God. I'm loving that. Thank you so much for coming and sitting with us and having a chat. It's great to get people like you on this, and we'll kind of go more into who you are and what you do. But we're here today to talk about basic biology of lung cancer and then about oncogenic drivers. We will explain that to everybody. I'm just wondering if you could start by introducing yourself and a little bit about what you do and what your day-to-day is and the life of Colin Lindsay.
Yeah, of course. And, there's so many different ways you can think of this, but I guess technically my term is a clinical senior lecturer for my job title. Okay. My split between research and the clinic is 50-50. In the clinic, I mainly focus on lung cancer, and as I guess we'll discuss later on, in terms of the research focus, it's very much on this gene called KRAS, and there are 3 different types of it. I maybe can say early. So KRAS is just one of the RAS genes. And it's the most commonly mutated oncogene in lung cancer and cancer overall. So that's where my research strategy is really focused.
Fab. And just in terms of the clinic, where's your clinic?
My clinic is at the Christie NHS Foundation Trust, the Christie Hospital. I personally lead 2 clinics a week on a Tuesday and a Friday, with mainly with the lung cancer team, but also our research team as well, as far as clinical trials and sample collections are concerned.
Fab. And the Christie is obviously the biggest cancer hospital in Europe, so it's an amazing place to work, I'm sure.
It really is, and you can tell I'm not from here, but it was such a pull to come here. One of the big reasons I came here was it is, an internationally dynamic lung cancer centre. Yeah. But also, I think maybe it being elsewhere kind of helps me appreciate as well having the labs. Normally all in the same site as the hospital makes such a fundamental difference to what you can do for translation. Yeah,
it's that cancer campus idea, isn't it?
Yeah.
Oh, fabulous. And within your role, so you've explained that you're kind of in the clinic and your research, what are your main responsibilities? What are you accountable for?
Well, I guess if I start with the clinic, I guess first and foremost, the patients and the patients' lives. So I'm responsible for making sure they're on the right treatment, that it happens on time, that we're making the right decisions about when to pause things, when to reduce their doses, when not to treat. And everything, I guess, really comes down to some level of risk balance. And I think I'm responsible for being honest to the patients and not glossing over what are often very difficult situations. And so that would be the first bracket of things. In the clinic and crossing over into the research, the second part of things is making sure clinical trials run properly. So I'm the chief investigator, which means that, the main lead in the UK for a number of different clinical trials. Many are related to this gene called KRAS. We need to make sure that the governance and all the paperwork and all the ethics of of those studies are carried out responsibly. Again, crossing over to the patients, making sure that we're as transparent as possible in terms of what we can achieve and what we can't achieve. And then the last main bracket of what I'm responsible is the research that we do beyond the patients, and that's preclinical research. So I supervise, I think, 3 PhD students and maybe 1 or 2 postdocs as well, who all lead projects that are linked with some of the samples we try and take from the clinic, so that we're really trying to use the samples from the clinic as a basis for our research and driving the questions, rather than probably the traditional model was to do it the other way around.
Yeah, so it's really like bench to bedside, bedside to bench in that translational manner, which is so impressive.
That's and it's really, really difficult to articulate how difficult that is to achieve, and it's quite boring to articulate it as well, in terms of making sure all the applications are right. But it is a difficult thing to achieve, and the infrastructure in Manchester is really second to none in helping with that.
Yeah. So if you guys have listened to season 1, you'll have heard the research journeys of some of our other researchers. I don't know if you've had a chance to listen, Colin.
A little bit, yeah. Not all of them, I must admit. But
all you guys are in the right place, because you get to listen to Colin now. And I mean, if you haven't listened to season 1, go and listen. I personally think it's great, but I also host it, so might be a bit of bias there. But I mean, my next question would be that kind of personal, like, how did you become involved in cancer research? You can take it as far back as you want, maybe if you have a story from when you were a kid, or
probably not. I'm not one of those kids that knew, for instance, that I wanted to do medicine from an early age. It was a sort of late realization to some extent. What it brings me back to is my general medical training. When I was very much a junior doctor, still in my mid-20s. And I worked in Glasgow at the time, and I worked in a hospital called the Glasgow Royal Infirmary, which covers the east end of Glasgow. It probably, like many areas of Manchester, and there are many similarities between the two cities, it covered a lot of deprived areas in particular, and there are constituencies in the east end of Glasgow where the average life expectancy falls short of retirement age, quite a few of them. And so for me, being on the shop floor in receiving what I would see very, very commonly was a patient who would come in breathless or with a cough for the first time. They should have probably been to the doctors months ago, but for various reasons they hadn't done that. And so this was their first presentation. We would do a chest x-ray and we'd see that they likely had a metastatic lung cancer even without a biopsy. And so I think, a lot of it comes back to those sort of patients, and being able to offer them something, because by virtue of the fact that they came to casualty, we knew that they were probably too frail to actually receive chemotherapy, which was our mainstay of treatment at the time. So what we needed for those patients, and we still need for those patients, is a treatment like chemo, yes, that works quickly, but it needed to be a tolerable treatment as well. And that's what we couldn't offer at the time, and we're only just beginning to realise, if I'm being honest.
And I mean, it's crazy thinking about that I mean, people are coming into A&E and then being diagnosed with lung cancer, and I think it shows that, you know, it's late presenting in terms of its symptoms. So you're presenting kind of with that cough and that shortness of breath and maybe coughing blood and maybe fatigue. And that's, you know, on stage 4 of the journey of cancer where you get that metastatic rate. And it's crazy about that presentation. And we've kind of talked about like early detection previously, but it links in with that. And I mean, you flagged something as well about the East End of Glasgow and the deprivation. And I'm just wondering, correct me if I'm wrong, I think Manchester has some of the highest rates of lung cancer in the country. And I'm wondering if you could tell us a bit about the factors that drive that and the inequalities in lung cancer here.
Sure. And I mean, like most people, I do sort of refer to CRUK statistics intermittently for this sort of information. And I think probably Manchester is slightly better than what it was, but ultimately it's still top 5 in terms of lung cancer incidence across all the different regions across the country. You know, the northeast is terrible as well. But given the sheer population in the northwest, I think it's reasonable to estimate that you know, the highest numbers of patients with lung cancer exist in this part of the country. And so this kind of brings me back to the other reason I wanted to get into it, which was it was a simple case of what can I do that will offer the biggest impact. 2/3 of lung cancer patients unfortunately present with advanced disease. A lot of the patients who present with early stage disease still unfortunately recur and become uncurable. And lung cancer is easily still the biggest cause of cancer mortality of all the cancers, about 20% of cases. And so in terms of wanting to offer an impact for patient well-being, this was clearly the place that I wanted to work in. I won't deny there are parts of it that really just bring in things that I enjoy, which we'll maybe talk on later in terms of the research. But it was certainly important to me to be centred in this cancer, which affects people's lives in so many different ways.
Yeah. And I mean, I think one of the rhetorics that we want to carry through this series that's really important to me, and I think it should be propagated, is that kind of the idea that you only need a pair of lungs to get lung cancer.
Yeah.
Do you? We understand that it's that like parts of it are a smoking driven disease, and there's other kind of cofactors such as, you know, Tracer X has been looking at pollution and what was it, like irradium or something. And I mean, do you see kind of smoking driven lung cancers, especially in the clinics here in Manchester? Because I know smoking's kind of one of the counterparts of social deprivation, and they kind of come hand in hand. And there are a lot of deprived communities here, as we know. So my question would be, do you see that translating into the clinic?
I think we definitely do, but equally because Christie is a tertiary cancer centre, a specialist centre. We do frequently see alterations, genetic changes that are more associated with the never smoking type of lung cancer as well. So we do get, I think, a very from our point of view in terms of knowledge, a very helpful mix that keeps up our knowledge and our expertise in all the different facets of lung cancer. But if you speak to my colleagues in the peripheral hospitals who are referring patients, they will tell you that it's mostly a consequence of deprived areas continuing in Manchester and the northwest, and smoking is inherently linked with that. And most of us get into lung cancer because we believe smoking is an addiction. It's not the patient's fault at all. They pay a heavy price in terms of their taxes, and so I think we try and offer the same level of care to absolutely everyone who walks in the door, and lung cancer is particularly fantastic because I sometimes say we've got patients that ask for nothing and thank us for everything, and they don't really question us too much, and so I find it really important to be their advocate, because sometimes they don't have as big of a voice as they should do. Yeah,
and I think that's one of the things, isn't it? Removing that stigma and removing the blame, because it's one of those cancers, I think kind of melanoma ties in, where there's that like causal blame associated with it, and it should just be completely ignored. There's no blame to it. It's an addiction. And like you say, I mean, I know there's been kind of in the past there were stigmas behind it and funding issues. You know, you get cancers like breast cancer that are massively funded because there's not that kind of causal link between something that you do and then you present with breast cancer. Whereas in lung cancer, in some of the cases, it is smoking driven. Yeah. But number one, that shouldn't be a stigma. And number two, in a lot of cases, is it something like 14%? It's people that have never smoked.
Yeah, and so that's right, and so and there and very understandably, people who have lung cancer that have never smoked are increasingly concerned that they're going to be stigmatized as well, and it is about I would normally just bracket it as 10 to 20%. I think it depends on the region you're maybe working in to some extent. And it gets a little bit more complicated as well, and so we need to be very careful in how we approach it because, as I'm sure we'll talk about, there are genetic changes more associated with never smoking related cancers. There are genetic changes that are associated with smoking related cancers, but there's also a grey area on each side where never smoking patients can actually still not infrequently have mutations associated with smoking-related cancers, and patients who have smoked in the past not infrequently have genetic changes associated with never smoking cancers. And so, if we're too strict with our terminology, then those patients that fall into that grey zone can potentially be disadvantaged.
Yeah, God, it's a minefield, isn't it? So, just taking a step back and looking at the basic biology, so. The way that I understand lung cancer is divided into those kind of two subtypes, one of which being small cell lung cancer, the other non-small cell lung cancer. And then within non-small cell, am I right in saying there's squamous, adeno, and large cell? Please, could you tell us a bit more about those kind of like derivatives of the cancer and how they're defined?
Yeah, sure. Of course. So there are different divisions in terms of this classification, and so the first thing, as you mentioned, is small cell and non-small cell, and that's the most fundamental part of what we look for, whether it's early stage or late stage disease. The small cell is as simple as it sounds in many ways. The pathologists are looking for little blue cells when they're looking at a biopsy sample or a liquid sample we call cytology sometimes. And that's about 10 to 20% of cases. So a minority, but a very important minority because it's usually the more aggressive subtype of the two lung cancers, which we've generally been able to offer less in terms of treatment modalities and survival benefit. Non-small cell is clearly the biggest subtype, and it can be broken down further. It's about 80 to 90% of cases we will see, because it tends to be slightly less aggressive than small cell, probably more early stage cases with non-small cell that can be cured. And beyond that, the things you mentioned, Sally, is that non-small cell can mainly be broken down into adenocarcinoma and squamous subtypes as well, with adenocarcinoma being about 40 to 50% of cases. We see adenocarcinomas in other types of, I would call epithelial cancers like colorectal cancer, pancreatic cancer. But these are adenocarcinomas we can tell through pathology are specific to the lung. With squamous cancer, it's about 10 to 20% of cases. They're actually more difficult to differentiate from squamous cancers in other parts of the body, which can cause diagnostic problems sometimes.
Yeah, God. Well, I'm glad that I've been doing my homework, at least. And in terms of who's most at risk of developing cancers, is that people with these things called oncogenic drivers? Or is it a variable?
Yeah, I think it's a big debate just now about what point in the cancer evolution those oncogenic drivers come on.
Okay.
There's an increasing body of research suggesting that they're there to some extent in healthy cells.
Right.
And so it's difficult to say that they are the cause of cancers. I don't think they are, to be honest. I think it's much more complicated than that. And that's where one big aspect of cancer research is rooted just now. What is that tipping point that leads a healthy cell to become a cancer cell? I don't think it is just one of these oncogenic drivers, but what they do clearly help with increasingly, and lung cancer is really the pioneer, the leader for this in terms of setting the example for precision medicine, as they allow us to prognosticate and offer treatments for those specific changes that can often be beneficial. For example, if you have an EGFR mutation in your lung cancer, we can give you an EGFR inhibitor, and that will be very likely to extend your life. And it's a tablet as well, so it really offers quality of life. We have an increasing number of these genetic targets that we're uncovering in non-small cell lung cancer in particular, and adenocarcinoma in particular.
Okay, so just back to the definition, are we just okay to tell the listeners what exactly is an oncogenic driver?
Yeah, I mean, an oncogenic driver. Oh, now you're putting me in this.
Go on, Colin.
I'm not ready for this one. An oncogenic driver gene is really a gene that either through population studies or preclinical work has really been thought to make the cancer cell signal in the way that it likes to do, driving proliferation of the cancer cells, so the cells dividing, allowing it to evade a cell death signal so that it survives better than healthy cells as well. And so that would broadly be, without my preparation, what an oncogenic driver does.
Wonder if you'd score 4 points for that.
Probably not, no. Yeah.
I'm giving you points anyway. So you've mentioned KRAS, EGFR. What are other kind of common oncogenic drivers in lung cancer?
Yeah, I guess many people might remember from some sort of previous schooling or training that they can be broadly split into oncogenes and tumor suppressor genes.
Yeah.
So first thing to say is we're mainly talking about oncogenes, and because clearly tumor suppressor genes are things that you wouldn't want to target with drugs. So I'll move them to one side, and then the oncogenes are the two main ones you've mentioned. So KRAS, which broadly is more associated with smoking, but as I say, we can still see it in never smoking patients, and EGFR, which again is what was the opposite, is more associated with never smoking, but we can still see it in smoking patients. Going beyond that, one very successful story is a gene called ALK, which is rearranged in lung cancer. We have and then we have almost a never-ending list at this point of genes that affect very small percentages of patients, such as RET1, ROS1. There's one called NTRK, which is NTRK. BRAF is another one with a targeted success story closely linked to RAS as well.
Well, there you go, everybody. Some genes there for you. Yeah, just on ALK, we're actually going to be speaking to Fabio Gomez, who is doing ALK positive research now, looking at the ALK gene in never smokers. So very exciting times ahead. So you guys are in the right place now, but stay tuned, please, because I have so much more coming. And so you've said that you target treatments towards a cancer. Like I'd kind of say it's like a precision medicine. So you target them in accordance with if they present with a certain mutation. So we'll get onto KRAS because it's, a big bulk of this podcast. Don't want to give you spoilers too early. But in terms of KRAS, you'd kind of target that with a specific kind of drug to target that mutation. Just taking a step back, how do you test a lung cancer for a mutation? Like, how would you pick up if it has EGFR or ALK or KRAS?
Sure. I mean, the first thing to say is that we need to get a good biopsy specimen, and that is very challenging. And there was a study just in our main international cancer meeting, ASCO, 1 or 2 years ago, that in terms of achieving a good enough biopsy sample to see a full biomarker readout is still not being done well enough, even in the States. And so it was less, I think it was less than 50% of patients that were managing five biomarkers. And so I've already reeled off, I haven't even mentioned PD-L1 for immunotherapy, but I've already reeled off a list of, I think, about 67 genes that are important for people. And so, and the challenge with that is that we have an increasingly great level of technology that can diagnose patients with cancers at distal points of the bronchial tree. But of course, to do all these extra tests, we need as much material as possible. But once we're getting to these distant points, it's very hard to achieve that. So I would say probably we're looking at a little bit of a future where a second biopsy is something we need to consider more and more, given the range of possibilities that can be offered in terms of treatment. I need to go back to your question.
Don't worry, it's just
I'm waffling.
How do you test for the lung cancer mutations?
So once you've got a good biopsy, right, I'm going to be brief this time. Once you've got a good biopsy specimen, basically just across the road, mainly in St Mary's, Manchester Cancer Genomics Centre, what we can do is various tests. Next generation sequencing, many people will have heard of, and that can test for many of the genes I've mentioned. We do have single gene tests still, such as PCR, when we need to focus our testing, and then we still do immunohistochemistry and FISH testing as well. Okay,
and is that routine for before lung cancer treatment begins, or is that something that you're trying to introduce?
There are a lot of ongoing conversations about this. Most of the targeted treatments and biologicals are rooted in treatment of advanced disease, which we've said is the majority of patients. But increasingly, we're bringing systemic treatment and immunotherapy into early stage disease as well now. And it is important to do this testing in early stage disease, we've realised. And so I think it's something we're going to be offering to all patients. Yeah. It's actually easier to offer it to earlier stage patients because they're likely to have surgery. And so we know we're going to have a good specimen.
Yeah. Yeah. Interesting. So, time to shine, Colin. We've mentioned KRAS.
Oh no, I put pressure on myself here. Oh.
No, because it's what you do every day. I've tested you with oncogenic drivers. Sure. It's like going back to A level for you, but now this is your PIP and present. So, in terms of KRAS, could you tell me a bit more about this specific mutation, and then we'll kind of like feed on to the work that you've been doing and what have you?
Yeah. RAS was discovered as an oncogene over 40 years ago now. And so really during my PhD, where I worked on the gene next to it called BRAF, I knew that the BRAF mutation had been discovered and a drug had been generated for it very, very quickly within the space of about 5 to 10 years. And that BRAF inhibitor was showing success in the melanoma clinic. Very nicely in offering quality of life to patients. Arguably, the survival benefits could have been better, but unquestionably, it was helping patients. So knowing about RAS being there for 40 years, and also knowing that it was the most commonly mutated oncogene in cancer, in lung cancer as well, I became really fascinated about the possibility of drugging it and why that hadn't happened yet, and wanting to understand that. And it does come back to this thing where I wanted to also offer the biggest impact. And like I say, it was so common in patients that I just wondered how transferable that BRAF example was. And the answer is it's extremely difficult to drug RAS, and that's why it's taken so long. Perhaps not a surprise. Yeah. And the big difference between RAS and all the other targets we tend to talk about is they are enzymes fundamentally, if you go back to school chemistry. But RAS is a different type of enzyme called the GTPase, whereas RAF and many of our other targets have kinase activity. And RAS binds much more tightly to substrate GTP. then kinases bind to their substrate ATP. So it's very difficult to find drugs that will interfere with that interaction.
Right, okay.
There is a second thing, Sally, which I can just be brief on, which is that it's actually a very small, smooth protein.
Yeah.
So it's as simple as just thinking, well, is there a pocket or a little hole in the protein for a drug to cling on to? And that was something that was very difficult to find before.
Yeah. God, really sending you all back to GCSE chemistry and biology. God, it's sending me back as well.
Chemistry, I think it's biology.
No, there's chemistry in there as well. We see this in never smokers as well. Just looking back, so this is a non-small cell lung cancer that that KRAS is found in, is it? Or is it found in both?
Mainly in adenocarcinoma, like most of the targets we're going to talk about.
Okay.
But it accounts for over 90% of pancreatic cancers, which is, if anything, has an even worse outlook than lung cancers, you probably know. And it accounts for about 40 to 50% of colorectal cancer. So making impact in this space is a huge thing for patients.
And then is there NRAS in melanoma?
There is NRAS in melanoma, very good.
I've done my homework.
So I don't know if I have a percentage available for you, but it is a very large, I think it's less than 50%. It's maybe about 20%, I think, of melanoma cases. And
then in never smokers, I think I read, is it like 2/3 of the never smokers that present with KRAS are female? So there's also that difference between the male and female presentation. I guess you don't know why that is yet. Yeah,
so there's more females with a KRAS mutation, and undoubtedly, and so I don't think we fully understand why that's the case. It is. We should say it is also a mutation that seems to be fundamental more to Caucasian populations. Whereas mutations like EGFR, we know, are far more prevalent in Asian populations.
Yeah, God, it's interesting, like the population level of it. Looking at KRAS. And the mutation in terms of survival of a patient, is there any kind of like empirical evidence that shows that it affects, somebody with a KRAS mutation has a lessened survival rate than somebody without it? Is there any kind of research on that at the minute?
Yeah, that's a really good question, because I've always sat at this junction of scientists and clinicians, and 5 years ago when I started here, we hadn't seen any drugs come through for KRAS, and I was telling people that they were coming, and a lot of people, I think, in the clinic didn't believe me. So, and the mismatch was such that scientists, I think, are a bit reluctant to get involved with RAS because it's such a competitive area. They see such fundamental differences in all their models that the research output is very high in the preclinical space. But for clinicians, I always struggle to get them interested, because, and I could see why. Ultimately, there weren't many studies that really showed clearly that it offered a prognostic difference to patients compared to cancers that didn't have the RAS mutation. And also clearly at the time, and this has changed, we didn't have any drugs available for RAS, and so that is the bread and butter for oncologists. And so there wasn't much more to say as far as that was concerned. The one place it has had a clinical impact for a number of years was actually as a negative biomarker.
Okay.
So if you had a colorectal cancer and it had a KRAS mutation, that would tell some of my colleagues in that space that an EGFR inhibitor wasn't going to work. And the reason for that was the EGFR inhibitor was blocking upstream to the KRAS mutation that was just turning the pathway back on again.
Okay, that's so interesting. And is there, do you know about any factors that, does the KRAS mutation affect metastatic rate at all?
Yeah, I wish it was clear cut, but this probably comes back to that point I made earlier about it's not, I have to admit that it's not just about KRAS and tumor initiation.
Yeah.
And in the later stages of cancer, I also think it's not just about KRAS, but I do think, ultimately we need to choose our targets wisely and our where we focus our research wisely. I do think for so many reasons, RAS is such an important space to focus in on. but the studies that look into prognostication, for instance, with KRAS mutation versus no KRAS mutation, different subtypes of KRAS as well, which we'll maybe talk about in terms of the drugs. They offer conflicting stories, and I don't think they show a clear line of evidence that if you have a KRAS mutation, your prognosis is worse in most cancers, and that's because other factors come into play. I'm sure you'll have experts in talking about all the different facets of cancer biology that will tell you, about all the layers of things around the KRAS mutation.
Yeah. And the message today, people, is things are never as simple as you want them to be.
That's definitely true.
That is what we're carrying through. So, I mean, just in terms, I'm going to really summarize this in a lay way, but you've previously said people thought KRAS was undruggable just because it had a massive affinity to GTPase, and it was hard to kind of get a molecule in there that would have a greater affinity to, and it's also a really smooth protein, so it's hard to find a binder for that. Right, okay, cool. So leading on from this, I mean, you have looked into treatments for KRAS, this undruggable mutation.
Yeah, so that's a good lead. Thank you. It's druggable now.
Yes,
and it's lovely to be able to say that. I used to do a talk entirely on the disappointments with KRAS targeting in the past, and now that's just one slide of my talk, my sort of standard talk that I give to audiences. We have many drugs. They are for a specific, and this is how precise we've had to be with precision medicine. Arguably, these are the most precise drugs we have available. We have offered to us now, and like I say, they're not kinase inhibitors. Technically, they're GTPase inhibitors, but I guess we loosely refer to all these drugs as small molecules. And the point mutation of KRAS is the most, just by luck for me, is the most common one in lung cancer, which is G12C. So you take codon 12 in the gene and you swap a glycine amino acid for a cysteine. Right. And so we have other, we'll maybe come on to this, we have other amino acid changes such as glycine for aspartic acid called G12D. These drugs available just now will not work even with that one amino acid change. And so that's what I mean by the level of precision they offer. And so they have shown the G12C inhibitors, as I'll refer them just in case we start talking about other mutations. They've offered clear activity in lung cancer, colorectal cancer, and pancreatic cancers that harbour this G12C mutation. Unfortunately, it's not as common in pancreatic and colorectal cancer. It's broadly associated with smoking, the G12C mutation, which is why many people assume it's common in lung cancer. It's about 10 to 15% of cases.
Okay. And guys, no worries if you don't know about all those amino acid sequences.
Sorry,
No, don't apologise.
If I'm getting too technical, let me know.
It's fine. It's just a very complex landscape that we're working in, but I will do my best to translate.
I would normally have slides next to me, so
don't you worry, Colin. Please, could you tell me about sotorasib? Is that right?
Yeah, very good. Yeah. And the Americans call it sotorasib, accentuating the ras. But I think it's a nicer way to say it, sotorasib. It's less severe. Anyway.
Until we get onto linguistics.
Sotorasib really was the first G12C inhibitor on the clinical scene. I should say that, the development of these drugs was first and foremost an academic drive. And so it was work from a chemist in the University of California, San Francisco, called Kevin Shokat. And he had a lot of discussions with a guy called Frank McCormick, who's actually an Englishman who unfortunately departed England, I think many decades ago now. And Frank basically encouraged Kevin to focus in on RAS with his chemistry expertise. And that's why we have these drugs. What happens is they publish the chemical structure, and then pharma absorbs all of that and creates their own slightly different drugs that fundamentally, I think, mostly do the same thing. And so sotorasib is the first example of a pharma iteration of these drugs. And it's produced by a company called Amgen. And for balance, adagrasib is also a front runner, but came slightly behind sotorasib and is showing good clinical trial success as well. And adagrasib's made by a company called Mirati. So sotorasib we've looked at in lung cancer mainly in trials called the Codebreaker studies. And what we published recently in the Lancet with Manchester, leading this in the UK, is the Codebreaker 200 study. What the Codebreaker 200 study has shown is that in patients who are due for a second treatment for their advanced lung cancer or metastatic non-small cell lung cancer with the G12C mutation, that this drug is better than our previous standard of care called docetaxel. And by better, you can interpret it in many different ways. One big thing clearly is it's a tablet. It offers convenience for patients and I would say far less in the way of side effects and risk. But there have been one or two eyebrows raised about the level of efficacy or activity it offers, and it isn't synonymous with what we've seen with tyrosine kinase inhibitors yet. I can probably say that probably the best metric that I can say for the Codebreaker 200 study is that at 1 year, about 25% of patients in sotorasib, their cancer wouldn't have grown still, but with docetaxel, it was 10% of patients. So 90% of patients in docetaxel, the cancer would have started growing again by that 1 year landmark.
Yeah, and just for you guys, we will put the links to these papers in the show notes because it's very useful to look at them. I probably will read them a lot more than you guys do, but that's okay. And so when was that what was it called? A Codebreaker?
Codebreaker 200.
God, it sounds like some kind of like 007 mission. Colin, you going on the Codebreaker 200?
I know, I never thought about it that way, but these acronyms are getting out of control. What can we say?
I love a science acronym.
We've got a whole series of clinical trials that are dedicated to butterfly acronyms. So we have, what is it? Chrysalis, Mariposa. I don't think we've seen Papillon yet, but that's just it's only a matter of time.
Yeah, there's a study going on called Cup at the minute, and they're like putting kind of like teacup or like egg cup, and that's like the kind of the way that they're naming it. It's bizarre.
I mean, we were at a lung cancer meeting on Friday, and I've got a colleague that works heavily in mesothelioma, and he'd called one of his studies Miso Origins, and now he realizes that everyone just thinks of it like another Marvel movie. And then with some of the biobanking work we've done with RAS, I called our study, and I think it's too difficult to change this now, RAS Bio, but now I just think of it as a sort of naff, dangerous yogurt brand.
RAS Bio. Yeah, don't get people onto that.
No, exactly. Yeah.
RAS Bio is not for your gut health.
No, exactly. Yeah.
But anyway, back to it. When was that study, the Codebreaker? When did that start? That hit off?
That probably started a couple of years ago, and the interest in these drugs is such that these trials are recruiting like lightning, and so it was the recruitment period was short. We were lucky enough to have patients particularly well with this study. But it was short, and one big thing we're focusing on is creating more opportunities for patients to access these drugs in the future. I mean, maybe if I can say, we have a number of trials with the potential to access G12C inhibitors ongoing just now, not just for lung cancer. What colleagues email me about frequently is the other KRAS mutations, and maybe I should say now that G12D is the most common RAS mutation in cancer in general. It's still pretty common in lung, but it's very common in pancreatic and colorectal. Yeah. G12V, which is a valine, it's actually the second most common. So G12C is more dominant in lung, but it's the third most common overall. And clearly the emails I get from colleagues is, when are you going to have drugs for G12D and G12V? I mean, I really want to be able to offer those trials to patients, and fortunately, I can say it's not just limited to the transferability of creating G12D inhibitors, G12V inhibitors. There has been this sort of follow-up now where pan-RAS inhibitors are being created. So we don't necessarily need to be completely restricted to these point mutations, hopefully in the future. The question of toxicity and side effects with pan-RAS inhibitors is going to be a big one.
Okay. God, it's a lot. I bet you're like, blimmin' hell, I've only just found this G12C drug. Calm down a second. Let me get this out.
Yeah, it's complicated and it's a test of the brain even when I get to focus on it most of the time. But it is, you know, it's great to see ultimately.
Yeah. So just taking it back a second, I'll try and kind of put this eloquently. Sotorasib was compared to standard of line treatment in patients with non-small cell lung cancer that had already had like first line, would it have been chemotherapy or immunotherapy?
Yeah, either together or one after the other.
Okay.
Yeah.
And then, so that was compared to Doxatal?
Docetaxel.
God,
getting there.
Thank God you're here. And then it was found that sotorasib was better in terms of the patient survival, but also reducing kind of toxicities and side effects for that patient.
Yeah.
Right.
It gets more complicated now. I'm not gonna go into the specifics. I don't know if you'll do it in another episode, but there's progression-free survival and there's overall survival. Now, the benefit wasn't shown for overall survival, but there's good reasons for that in terms of methodology that are difficult to that are difficult to normalize for. But the progression-free survival benefit was there. So
progression-free is just like no tumor shrinkage, but it's not metastasizing or growing.
So progression-free survival is time till tumor growth or death, whichever occurs first.
Yeah. Okay. Cool. That clears things up. And I mean, where's Taurusip at now? In the FDA.
Also,
and nice.
Yeah, I mean, it's probably long approved by the FDA
now.
Yeah,
quite a while ago.
And adagrasib, I think, is just there as well. Now, adagrasib isn't NICE approved, and it's purely sotorasib in the UK, but we are doing better than some of our brother and sister countries in Europe. I know that Spain doesn't have access to any KRAS inhibitors, which is a particular shame for them because they've got a really, really strong research base in RAS. But sotorasib is accessible for second-line treatment of advanced non-small cell lung cancer now if your cancer has a G12C mutation, which is fantastic, and it is much quicker than it used to be in terms of some of the government changes in creating availability of these drugs. The next step, and this is and again another key differentiator from tyrosine kinase inhibitors, is how well it combines with immunotherapy.
Okay.
Because the cancers that are driven by EGFR, ALK, and some of the other ones I've mentioned, unfortunately don't respond to immunotherapy very well.
Okay.
One of the big reasons why KRAS inhibitors, G12C inhibitors, are still rooted in the second line space is actually because most KRAS mutant non-small cell lung cancer can do very well with immunotherapy. So that's set a very high bar for these drugs to enter this first line space. And I think realistically, they're far more likely to do that if they combine with immunotherapy in some way rather than trying to knock them off the top tier, if you like.
Okay. We'll come on to that, definitely, because that's interesting stuff. And I mean, for the listeners that are kind of, long term, have been here since season 1. We spoke to Catherine Payne and also Emma Crosby about, sorry about that computer sound. Yeah, we spoke to them about kind of NICE and getting into NICE. So that's the National Institute of Health and Care Excellence. Don't know where the H went, but probably wouldn't have created such a nice acronym. And we talked to them about kind of Lynch syndrome and getting just kind of standards of care and things changed there. I mean, what was the process for you in terms of getting sotorasib into NICE and that approval process? How kind of were you involved in it? Were you on the sidelines? What
I have a brief answer to this, and mercifully, I wasn't involved in it. I wasn't involved in it at all. But I mean, it's just a submission from the company. But in terms of the scrutiny, I wasn't involved in it. Perhaps it's probably a good thing that I wasn't involved in it, because I wouldn't have been able to be very impartial, I don't think. And I probably mentioned, sorry, just sidestepping your question a little bit.
Don't worry, sidestep away.
I probably mentioned. Well, I did mention there have been one or two raised eyebrows about how these drugs compare with tyrosine kinase inhibitors, because they don't compare as favourably, and I think it's clear to say in terms of their activity. But I would defend them clearly in the context of what I've said, which is that it's taken so long and so much effort to come so far that I, for one, am really glad that they have a footprint now in the clinical space. But also, you know, this is a fundamentally different target, and because most of the patients we're treating have smoking-related lung cancer, it's a fundamentally different disease. And the potential for rewiring of a cancer cell, I think, it probably inevitably meant that they weren't going to be quite as good as tyrosine kinase inhibitors in this first iteration. I would stress that sometimes I refer to it as knowing your enemy is the absolute crucial part of this. And this is just the first set of drugs we're seeing in this space. With EGFR and ALK inhibitors, it certainly took second, third generations of drugs to really create that impact that was changing lives on a regular basis.
Even if you think about it, like 5 years ago, something that was classified as undruggable, like, it's a massive breakthrough, irrespective of how kind of the effects that you see from that. If there is an effect, however kind of minuscule or whatever, it's amazing that this has progressed so far. And congrats to everybody.
Oh, thanks. It's really not been me, but it's nice to be part of. I think that there are probably 2 or 3 different proofs of principle that we don't reinforce enough. It was the first time really we'd seen a small molecule like this working in smoking related lung cancer, which, as I say, I think is really a fundamentally different disease given the mutation burden that's accumulated in that cancer. I mean, there was a big worry as well that there's always a worry that preclinical models just aren't representative of patient cancers. And so was that oncogene addiction that we see in mouse models and cell models really going to be seen in patients when we treated with this drug? Well, we can say yes, maybe not as well as is the case with EGFR and ALK just now, but clearly there is a dependence of the cancer on KRAS still, and that's why these drugs are showing benefit in patients. There is another proof of principle, and it slipped out of my head. It'll come back to me.
We'll wait for it to come back to you, but I'll ask you some other questions before it comes back to you. So in terms of the next steps and working with NICE, you said something about kind of looking at the drug in combination. So looking at sotorasib in combination with other treatments. How would you see that kind of working? Like, what examples would you have of that? Yeah,
not easily is the answer.
Okay.
And it does, an added layer of complexity with toxicity comes into this. We had previous trials with tyrosine kinase inhibitors where they were combined with immunotherapy, and we saw quite severe toxicity and to some extent unexpected idiosyncratic toxicity, particularly affecting the liver. So those trials were mostly sidelined because it wasn't so important. As I said earlier, immunotherapy isn't so important to EGFR mutant lung cancer, ALK rearranged lung cancer, and so ultimately it's not been prioritized for future progress. Now, with KRAS, as I said, fundamentally it's important to try and combine these drugs, and more than any molecular targeted space of cancer, this is the space we're going to be trying to combine our 2 most exciting modalities of treatment as a medical oncologist: small molecules and immunotherapy. And the problem is that the initial indications are that we're still seeing that toxicity with KRAS inhibitors. And so that's been described to in particular in another Codebreaker study with sotorasib. there is a suggestion with the other drug I mentioned, adagrasib, that there might be less toxicity when combining with adagrasib and immunotherapy together, but we still need to see the data in that.
Yeah.
What I do know is that there are clinical trials definitely trying to combine the drugs in the first line treatment space for advanced non-small cell lung cancer, and inevitably they're trying to take precautionary measures like changing the dose of the RAS inhibitor, looking at different dosing possibilities as well.
Yeah. So just a step back, because I think this is kind of a question that's popped into my head now, and it might be relevant for earlier on in the episode. So in terms of first line treatment, that's when a person first presents and you have that kind of standard of care. So if they're, say, stage 1, you might have a resection, which is like the cutting away of the tumour in the lung, and then kind of following on from that, you've got that metastases. What kind of sort of treatment would you provide at the other end of things? What would be that first line treatment? Would it be like radiotherapy, chemotherapy?
I see. Yeah, no, I have to say, yeah, it's so complicated, all this terminology that is kind of second hand to us. But by first line, I'm really automatically talking about advanced disease.
Okay.
And so I just mean the first treatment once you've been diagnosed with a metastatic or advanced cancer.
Cool.
What you're referring to is probably called either adjuvant or neoadjuvant neoadjuvant treatment mostly. So neoadjuvant, when you get systemic treatment before an operation and have a cancer that's curable. And adjuvant, the treatment that follows an operation in an effort to still try and to try and add to the potential for cure already offered by the surgery.
Okay. And then the first line is for that metastatic. Cool.
Yeah.
And then for the metastatic, what treatments do they have specifically? So that would be what was given before this sotorasib and... Yeah.
I mean, our standard bearer really, and something that as I've said is going to be very difficult to supplant is chemoimmunotherapy, or in patients whose tumours look particularly receptive to immunotherapy. Sometimes we can actually, by using PD-L1 immunohistochemistry, just drop the chemo and give immunotherapy by itself.
Yeah.
And so Keynote 24 with pembrolizumab and Keynote 189 with chemo and pembrolizumab are really there are a number of trials now looking at different immunotherapies in this space, but really they're the two main trials that we follow with our treatment. Okay,
cool. Right. Yeah, it's difficult to understand kind of clinical terminology, but I do my best. Sometimes not very well, but it's good to have you here because you can, pick me up. So, I mean, in terms of next steps for this research, what have you got under your belt?
Good question. How to articulate this? I mean, what I can say we're driving forward here are probably three main channels of work. First, when I joined, I really wanted to make sure that we had the clinical trials to offer patients. And I mentioned that already, but I've also mentioned that we need to work harder in trials for G12D and G12V now, for instance. But what I can say is that we are the UK lead for most of the RAS selected trials that we get involved with. And I think I'd like to think Manchester is a recognised centre for RAS based clinical trials now, and we link in very closely with international colleagues who I think recognise it in that respect. So that's a nice place to start from. The two main facets of what we pursue beyond that are, and this comes really to my background, is that we do have wet lab PhD students looking at RAS research, and so they are the students, mainly medical oncologists just now, who are using their pipette every day. They're perhaps doing some mouse work as well, and they're looking at different stories in terms of future vulnerabilities for RAS. One of them is looking, for instance, at G12C inhibitor resistance. And that's beyond the other RAS mutations. I would say that's a big big thing that people are picking up now and seeing what they can do with. I mean, we haven't talked about resistance. Do you mind if I segue a little bit?
No, go on, Colin. You can segue.
But the resistance that's offered by the cancer cell to G12C inhibitors is really, really striking because of the sheer range of different resistance mechanisms we've seen. Okay. So we're getting resistance in the same codon 12 of the RAS gene, different mutations there. We're getting mutations in different parts of the gene conferring resistance. We're getting mutations in new parts of the gene we didn't see mutations in before because it's where the drug's binding. And we're also seeing mutations in downstream genes and parallel pathway genes. And so the analogy I give for this just now is like, it's like hitting the cancer cell in the stomach, and it's fighting back with its head, its arms, its legs, its handbag, and its family. It's coming not long after as well. And so that's a real challenge. And one of our PhD students is looking at that in great detail just now. We have another type of RAS mutation affecting codon 13, which is quite common in lung, a rarer mutation, but more common in colorectal cancer. And one of our students is looking at the differences with that mutation compared to the standard codon 12 mutations. And then we have, on the other side of things, we have bioinformatic projects. Yeah. And I work with a cancer evolutionary bioinformatician called David Wedge.
Love David.
You've met David before. Yeah. So we have a common PhD student just now who's doing a fantastic job really at trying to unravel the taxonomy and classification of some of this so that we can start with some more core fundamentals in terms of what we're trying to achieve.
Just looking back at your kind of, what was it? Your handbag and the hitting in the stomach analogy, that's the word. In this instance, the hit in the stomach, is that sotorasib or...
Sorry, I should clarify.
Don't worry.
I think, to be honest, it just feels a little bit unseemly, but really I should be I would probably, it's better to say hitting it in the heart, because we do like to describe it as the heart of the cancer cell, or the engine room, or whatever you like, but that's what I mean, hitting it with the G12C inhibitors. We have the American Association for Cancer Research meeting coming up in a couple of weeks. ... And so the pioneers of RAS cancer research based in the US, they will be presenting, I think, our latest updates in the first plenary session at that meeting, and that's the main international cancer research meeting. So all I would say is watch this space. I guess I'd be surprised if we didn't find out more about G12D inhibitors. We definitely will this year if it's not at this meeting. We will certainly find out about more. I think we'll certainly find out about more refined iterations of G12C inhibitors, given the speakers that are going to be there.
God, it's going international.
Oh, it started international, unfortunately. But yeah, I think I think it's just fantastic to see. It doesn't really matter where it comes from. And one thing, that's an unenviable position, but people are really interested in with here is just that we do have this patient base. It can't be underestimated that translational research is very difficult in a lot of hospitals in the States, because they all focus on a much smaller cohort of patients, whereas a lot of patients affected by RAS mutant cancer are getting channeled towards our team or my practice, and that means that it means it can be more chaotic, but it also means if we're organized, our potential is stratospheric in terms of what I said about taking these patient samples, biobanking, and make sure we do that reverse translation. Yeah,
so you couldn't do it without the patients, and that is the power of the Christie. And I mean, it's you've been great to talk to, and you're so passionate and kind of clued in on all of this stuff, and especially with KRAS and things. And I think my question to you would be the question that I ask everybody that comes on here, is just like, how does it feel to work in cancer research and know that, at the end of the day, you are balanced between the clinic and the research lab, But knowing that is all being translated to patients, and like clinical trials are patient facing, right? And that's a lot of the work that you do. So how does it feel to know that, you know, you're having this kind of benefit and converting that previously kind of undruggable rhetoric into, no, it can be drugged, don't worry, guys.
Yeah, I mean, it feels. I mean, I think what you're suggesting is it feels great. I do get.
Just tell us it feels great.
I do get the other question, of course, which is, you know, you see incurable patients for your practice, and so doesn't that feel terrible? And truthfully, you know, it's always the patients that teach you, and they are so impressive, absolutely all of them in different ways, and they never fail to. I mean, I'm not a person that's easily surprised, but it's really the patient examples that surprise me, because they're just so gracious with their time and their energy at a time when they've got far more important things to focus on, and so it really is a privilege, and that's all I can really say. You know, we're at the forefront of some research, but more importantly, you know, we're seeing these patients whose lives are totally disrupted, and they're opening up to us, and you know, we do our best to help them.
You all do a blooming brilliant job. I know that for a fact, so applause to everybody as a clinician like Colin. And how do you balance it all?
I hope, yeah. I'm not the best example in that respect. But I try. I think the key thing is that, you know, if you choose to be busy, you try not to impose your expectations on others, your own self expectations on others, and so I mean, you know, you tend to balance it all by, you know, working most hours. I mean, I think it's fair to say I try and at least give myself one day off a week from it all. I've got 2 kids. I've got a wife who's a consultant in North Manchester, and we don't have any other family here, so it is frantic at times. I mean, it's mostly great fun. I try and see the positive in all of it, but it doesn't mean to say that it's not. There are difficult weeks.
You'll never be bored though, with a frantic life. How old are your kids?
10 and 7.
Crikey.
Oh, it feels a bit easier than it did a few years ago, because they can do some things for themselves.
Just wait 3 years. Just wait till that teenage phase.
Well, that's it. Yeah.
Yours might be better than me though. And just like, how was it working in Manchester? I mean, you said previously that you moved from Scotland to here. Yeah. Scotland's glorious, Colin. I love Scotland. Yeah. Why leave?
Oh, I mean, this is a 2-hour conversation in itself, but I mean, I think I have to say it first and foremost comes down to work, and it does for my wife actually as well. And I don't think that my job would be able to be the same in Scotland as things are set up just now. But the people I've met here are just phenomenal people, and you know, there are so many similarities between Glasgow and Manchester that it really didn't take long to feel comfortable. One always grounding influence are the nurses around you. And the nurses we have in the Christie team, the lung cancer team, they're easily the best nursing team that I've worked with in my time, and that makes my life and the patient's life so much easier. And so you know, we try and get out for a drink a couple of times a month and have a laugh as well. And I think that's always, you know, a group of colleagues that you think of with those sort of questions. I think I'm hugely thankful to them, and they're so impressive in everything they do.
Yeah, as are you. But it's phenomenal to see the way that the cancer research landscape works here. And like you say, you can't forget the jobs that facilitate everything, because there's people at the kind of the back of everything that happens. It's the way of production, but and production of great research. So, yeah.
I mean, if I could make a pitch for them, I think there's still a big gap in terms of advocating for people without a voice in all of this. And so that could be the people on the shop floor in the hospital who just don't have time to go for a pee or have their lunch. But also it is the patients who have morbidity and die so quickly that they don't have time to be involved with patient fora and tell us their experience. And so focusing, it's clearly my research is focused on RAS, but giving all of those people an opportunity to speak, I think, has just got to be a huge part of the future.
For sure. Great stuff. Well, thank you everybody for listening. If this is your first time, I hope you enjoyed Colin and I having a lovely chat on the basic biology of lung cancer and KRAS and all the hopes for the future. If you've listened before, you know you're in the right place and you're staying, I hope. But thank you to the James Bond of KRAS.
Well, I guess there's a Scottish connection, isn't there? Yeah.
Definitely. And I really hope that you've enjoyed your time. It's been great to have you on. It's really good to hear about everything. And from kind of such a personable person as well. As usual, guys, I'll link everything in the show notes. If you want to have a bit of a read around, I'm sure Colin will provide us with some kind of articles as well. But there's a lot of background reading if that's what you're interested in, and a bit of a nerd like I am. Nerd's probably a derogatory statement, isn't it? But geek.
Not at all. No.
We love science nerds here. But thank you guys so much, and we will speak to you next time. If you have been affected by anything you've heard in this episode, please see the show notes for a list of charities and organisations that can help.
1 in 2 was brought to you by the University of Manchester and the Manchester Cancer Research Centre. Listen to our next episode to hear from more of our researchers as they share the innovations, discoveries and projects that are changing the landscape of cancer prevention, early detection and treatment. To find out more about what you've heard today, please see the show notes for this episode, where you'll find a transcript and links to further information and research. Cancer is one of the university's 5 research beacons, showcasing the interdisciplinary collaborations and cross-sector partnerships that are tackling some of the biggest questions facing the planet. To hear more about Manchester's research in advanced materials, biotechnology, cancer, energy and global inequalities, go to manchester.ac.uk/beacons.